Hi, very nice talks. I'll have two questions, one for Julie, one for Christina. Maybe for Julie first. Could you talk a little bit about how, out of those 80% of the genes that you knew, how you picked the ones that you did, and particularly the HSP PB8, and what the process is from moving on from there? And then for Christina, maybe you could speculate about why the PPS had the differential effects both in vitro versus in vivo, and could it have something to do with dividing versus non-dividing cells, and could it be dangerous as well as beneficial based on the human data that there is? Some show no effect, yet some showing that the longest living variant CJD patients had gotten the PPS.
Okay, so the way that I analyze the data is basically I took the changes that were happening in the cells that were elk expressing cells, deer expressing cells, and ovine expressing cells, and basically compare. They all have multiple genes. Their list would range from anywhere from 800 genes to 1,000 genes. And when I say that, I mean when I compare resistant cells versus sensitive cells, the differences between those two cells, I would get a list of about 800 genes. I would then see, well, what similar genes are changing in the deer, similar genes changing in the elk expressing cells, and similar genes happening in the ovine. That's like kind of the basics. And then whatever is happening in the same, so whatever is similar in all three groups, that's where I got the 136 genes, or 108 that are known. And when I say known, they know the sequence has a protein function. Does that answer that question?
No,
Okay.
sorry, I guess you didn't understand. So my question is, how did you pick? I understand how you narrowed it down. You saw the differential expression,
right?
But then you picked one. Why?
Yeah,
why
no,
that
and
one?
I think I picked one so then once you get those list of genes, you can then put them in pathway analysis. And when I did that, HSBP8 kept coming up as being important in neurodegenerative pathways, as well as it was a chaperone, which we know chaperones are important in helping proteins fold correctly or dealing with them when they're folded incorrectly. So I knew that HSBP8 probably could be interesting because of the way that prion diseases are with their misfolded protein. And then the more I looked into it, I discovered that it also acted on the similar pathways that we had found earlier with the EF2 alpha phosphorylation to be important. So I knew that it was some way playing on a role on a pathway that we already knew was important, but it's just upstream. It hasn't been studied in prion disease yet till now, I guess.
But you might go on and study some of the other as well, yeah.
Right, exactly. So the plan is not just to stay with this one candidate. The plan is to screen as many as I possibly can. And like I said, there's six other genes that play a role on this same protein synthesis pathway that are being found in the screen as well. Some ribosomal proteins, which are important in protein synthesis, as well as some inflammatory markers or inflammatory pathway that can also lead into the same kind of pathways. So it's really interesting. I just, out of the interest of time, just showed this one candidate.
But I think the general point is if you do something like this, you end up with a lot of different possible candidates, and it's really hard to select what the appropriate one is, or the fundamental one, isn't it?
I think the advantage to our setup, too, is that we're using multiple different types of prion diseases in a sense, because you have the deer and the elk and the sheep, and the plan is just to keep adding to that. And so it helps us narrow down a list of things that's happening in general prion diseases.
So the difference between what we see in vitro versus in vivo with the heparan sulfate molecules is really interesting and intriguing. One possibility that's been raised by Byron Coe is that the endogenous heparan sulfate molecules would be promoting prion conversion and acting as a cofactor for conversion in vivo. But then when you administer these sulfated glycans as a therapy, that basically they act as a competitive inhibitor, so that they would bind, instead of the endogenous molecules binding, instead you have these other molecules coming in and they bind the PRPC, but are not promoting conversion, so they actually are locking up and preventing the conversion on the cell surface, or potentially as PRP is shed from the cell, which occurs about 10% of the PRP is shed from the cell, it may be that these molecules are binding and just preventing that conversion process from occurring. Another possibility is that when you administer these molecules, it's actually causing the PRPC to be brought into the cell and may affect the targeting of the PRPC molecules. So it's not completely known, but those are some of the possibilities that have been identified at this point.
Okay, so Tom. Should we wait until we get so everyone can hear? Sorry, then
What part of the brain does CJD most commonly start? What part of the brain?
Well, I think the first thing is it depends on what form of CJD you're talking about. That's sporadic.
Yes.
You're asking which part of the brain it commonly starts in?
Yes.
Well, I think the difficulty is if you're talking about sporadic CJD in human beings, we're not investigating their brain in detail at the time when it starts.
Right,
so it's very
that's
difficult
true.
to say.
Yeah.
So I suppose the way that clinical neurologists approach this is by looking at the symptoms.
The symptoms.
And what you see is that sporadic CJD presents in very variable ways. So some people present with visual disturbance and blindness as their first symptom, and presumably the disease is worse, therefore, in the part of the brain that deals with vision. Some people present with unsteadiness, and presumably it's in the part of the brain, the cerebellum, that deals with balance. And some people present with other features, and we don't know why. The implication is that in sporadic CJD, the disease appears to particularly affect certain areas of the brain in maximally to begin with, varying from individual to individual, and it's not entirely clear why that is. One explanation is that if sporadic CJD is a spontaneous disease because there's some event that occurs and you get protein conversion, it has to occur somewhere, and in one person it might occur by chance in one place and in one person another. I don't know, do you want to answer anything about that, Jean-Phillipe? Okay.
Of course, you have different explanations. One is the endogenous origin. The other one is the environmental one. And as you have multiple prion strains, depending on the interaction with the host, you can have multiple combinations. And we see it in animal models, and it's striking, and it's like that you can isolate different prion strains. And what people are saying now, we are speaking of one prion strain, but finally it's a cloud of variants. And depending on the way it is passaged, you are going to favor one or another one. So I think that since the beginning we say that there is a balance between the host with all the genes that can counteract and with the strain. And we go more and more in this direction. One fascinating thing which is occurring now is coming from yeast prions, where they are saying that finally the prion strains that we consider to be completely outside our field because it's not the same protein, it's another phenomenon, are counterbalanced by anti-prion mechanisms, and it's the balance between the prion mechanism and the anti-prion which makes appear or not the phenomenon. And if you have an increase in the protein which can convert into yeast prions, then you have to balance it by increasing the anti-prion mechanisms, which are chaperones or other things. And that is naturally occurring into the cell. If the same phenomenon are occurring in humans or in animals, it implies that prion phenomena would be more distributed than what we expect, and that's why we are apparently resistant to many things and to many other microorganisms. And depending on your heredity, you will be more sensitive to one thing or to another one. So it's not black and white, it's a balance.
I mean, certainly what I was talking about in the random change it's because it's thought to be an endogenous, spontaneously occurring disease. I mean, one other suggestion, of course, which relates to this, is that sporadic CJD isn't, and that it's acquired, and it's acquired by mechanisms that we don't understand, and therefore there may be different strains of infection, but we don't know.
I have a question for Jean-Phillipe about this very, very impressive mini brain. So have you checked the other cells in your mini brain, like microglia and astrocytes, because they're also very important in your cells, right? Yes.
Apparent.
The second question is for Christina about whether you are going to check also the PRPC, like those glycan, the difference, right? The different brain areas.
Apparently, mini brains are able to create all the subpopulations which are existing into the brain, and you have even plexus choroids which are there, or hippocampus, etc. So, and it's here that it's a little difficult because each mini brain is unique. Because, as I told you, the overall structure of the mini brain is not fixed. It's and depending on the mini brain, you will have different view, and that's why we envision to have an acquisition in 3 dimensions of the whole mini brain to, after that, be able with high computing, be able to standardize all this. So we are winning because we have more complexity, and we are losing because it's highly complex.
So thank you for the question. Yes, we do plan on looking at the endogenous heparan sulfate molecules that are also binding to the normal PRPC form. So what is that normally binding to? And also, does this heparan sulfate molecule differ between neurons and astrocytes and between different neuronal populations? I think that would be very interesting. Yeah,
Okay.
Dr. Moreno, are you figuring out what genes are turned on and off in your different models by extracting the microRNAs from the cell?
No, we haven't actually done any of that, although it's a very good, that's kind of the next way people in laboratories are starting to look at microRNAs. They're kind of they were a mystery a few years ago, but now we're starting to understand them a lot more. So it's definitely something that we would probably eventually start doing, but not at the very moment.
So I was wondering if you had any opinion or could speak to implications for CRISPR gene therapy in specifically transgenic GSS.
Okay. So yeah, the CRISPR technology is what I'm planning to use in the mice, starting with HSBP8. So what CRISPR technology allows us to do is to knock out the gene, or knock out the gene, and we can actually do it in our transgenic GSS mice, and we can do the whole brain. So that will be really interesting to do it that way. So yeah, and I think that that the advantages to CRISPR is there's supposed to be less side effects to knocking down other genes, which is when you start to mess with genetic knockdown, you can have other problems happen. So this is supposed to, although it's really new in the field, supposed to decrease those side effects. So we'll have to wait and see what happens with that. But
maybe I could ask you, Julie, a stupid question, because I'm not a scientist, but you said there were 8 unknown genes. Yeah, I thought that the gene had all been mapped. Am I wrong?
yeah, so I guess when I said there's 28 unknown genes, meaning they don't know what their function is. So they know the sequence, but they don't know why they're there.
So they're known genes, but
they
you
don't
don't know
even
what they
really,
do.
and they don't have names.
Yeah.
So they're just sequences of DNA.
So if you don't know what they do, maybe they could be important.
Right. So that's my plan. Yeah, exactly. So that's one of the first things I'm going to start doing with those is you can look at the structure to see if they look like any other genes. So like, do they have the same domains of a gene that functions a certain way? Then that can lead you down the path. So it's basically just a huge puzzle of DNA sequences that you have to try to figure out if they look like each other.
And I suppose another naive question, if no one else has got any, is you obviously very sensibly looked for the genes that were common to all, but could it be that there are genes which are responsible and important for each individual disease that won't necessarily be common?
Yes, definitely. And that's our second question, I would say, is that if, for instance, if you look at deer and elk, their brain pathology is different, so there's a reason. However, at the DNA level, they're only different by 1 amino acid, so only 3 of their DNA bases are different. And so it's really interesting that they have a different pathology, and hopefully this data, once we can get there to analyzing it, it might have an answer, but I think that's just the second question.
Yeah, sure. Yeah, so it's very complex. Yeah. So I'll ask another question about mini brains. They're sitting there with a cellular organisation. What are they doing? Do the cells communicate with each other then?
Nobody told them that there was no interest to do that. But you know that it's a little as an origami. These cells have the incredible capacity to self-organize. Yeah. And we suppose that if we had the possibility of sending the vascularization and there are signals from the whole body, they would go much more further. Yeah. So it's only that they are stopped in their developments because they are lacking things.
Yeah. That's interesting, isn't it? Yeah. Oh, wait, we have a question.
Yeah, this question is for Julie. You mentioned proteasomal degradation is one of the pathways for the protein of interest. So proteasomal degradation are usually ubiquitin dependent. So I was wondering if you found in your analysis other components of the ubiquitin pathway, like enzymes E1, E2, or some other conjugating enzymes.
Yeah, we actually, one of the enzymes, I believe the first one, UGT1, is found to be changed in my screen. So that's definitely where I'll take that direction when I look at proteasomal degradation.
So the change is upward?
Yeah, it goes up in the sensitive cells. So I would expect, that would be one of my next genes to go after as well.
So I suppose I could address this to all of you. If you're looking at the pathogenesis of disease and you're showing all of these extremely complicated interactions and genetic elements, what are the chances of finding a particular pathway that leads to treatment?
That's because I'm kind with you. Well, as in medicine, I learned humility. So when I began science, I thought that I would be very lucky if with one brilliant idea at the beginning and one pathway, I would explain everything. And that's why I've been so interested by mini brains, because we don't understand what they are doing, but they are doing it. Yeah. And we try to feed them now and to oblige them to infect, to be infected, and after to be treated, of course. So, as you know, with the aspirin, that's correct, we have been many, many years before beginning understanding what it was doing.
Sure.
But we were using it, and it was very helpful. Yeah. And we hope that with the screening test, we will be able to target drugs, even if we don't understand at the beginning what they are doing. But as this will be simplified models compared to the whole brain, then my colleagues will be very happy to test the drugs.
I suppose your comment about aspirin is quite interesting because, of course, we did use drugs before knowing how they worked.
Yes.
And it's quite complex sometimes how they work. So trying to find drugs by studying mechanisms isn't always successful, is it?
And we have a splendid explanation with Alzheimer's. You know that all the treatments failed, and we think now that the initial hypotheses are completely wrong.
Yeah, sure. Yeah.
So, and that's what we face regularly in many diseases.
Yeah, sure. So, question. Oh, so
I was just going to comment on that I think the field is taking multiple approaches towards therapy.
Yeah, exactly.
So. You heard this morning about approaches using antibodies as a therapy. There's a possibility of using molecules that downregulate PRPC expression, competitive inhibitors to bind PRPC, ways to cap the fibrils and prevent aggregation. So I think there's many strategies that different researchers are taking, and it may be that a combination of treatments is used in the end.
But just to, on the one hand, to reassure you that people are looking, but on the other hand, maybe you get some idea of how difficult it is. Sorry, one question. Yeah.
Okay. Just a follow-up question about the mini brains and using them for screening. As you weigh the sort of complexity, the uniqueness of each one, and the need to do such sophisticated imaging to account for the variability, what scale of screening is currently feasible or imaginable, and what do you see going forward?
Officially, if you take the actual technique, and if you are doing some mini brains, you are already exhausted, because what I showed you was slicing of mini brains, so immunohistochemistry and all these things, which are heavy, very heavy processes. If you change your paradigm and that you go towards an automatic high output screening, it implies that you are able to have an acquisition of the whole brain without slicing it, which is now possible. You are perhaps heard about clarity and other techniques like that, which allow to clarify the brain, and then you can have potentially an acquisition of the whole mini brain without slicing anything. And that's what I began to show you in the process. So we have the different bricks. It doesn't work for the moment, but I hope that when we will have finished the first process, the other part will work too.
Okay, so we have to finish, but I should thank our three speakers for presenting their work clearly to you.